Morning to our dearest chief guest and very good evening to all other participants. On behalf of the Department of Botany, let me extend a hearty welcome to the distinguished guests and all participants to the third lecture of the webinar series on Integrin Biology. The Department of Botany, University of Kerala, established at Thiruvananthapuram, the capital city of Kerala, has always been an academic and research focal point in the country. A city of tradition and space technology, Thiruvananthapuram truly represents the contrast India is well known for. The Department of Botany was established in the year 1959 by late Professor Dr. A. Abraham. Along with a galaxy of eminent professors, Dr. A. Abraham was instrumental in discovering the highest chromosome number in Ophioglossum reticulatum. The department also offers unique post-graduation courses in genetics and plant breeding and biodiversity conservation. Master of Philosophy in Botany is also offered by the department. The major research areas include cytogenetics, systematics, tissue culture, phytochemistry, seed physiology, and much more to add. A Center for Biodiversity Conservation is also exclusive for our department. In remembrance of Professor Abraham, the department publishes the journal Abrahamia, which covers research articles of utmost importance. The Haridalayam Project, a government initiative under the University of Kerala, endemic plant conservatory, etc., are the ongoing projects in the department. With this short note, may I invite respected Professor Dr. T.S. Sobna to officially welcome the guest as well as all other participants to this function. Thank you.
Thank you, Vinida. Most respected speaker of today's program, Dr. Yoshikazu Takada, dear colleagues from my department, other departments of our university, and other universities, faculty members, research scholars, and my dearest students. Good evening to all and good morning to Dr. Takada. It is indeed a great moment for the Department of Botany since we have a great personality like Dr. Takada is here with us. As most of you might be knowing, Kerala is in southernmost tip of India, and University of Kerala is a pioneer in the field of education and knowledge and is a mother university of Kerala from which many universities were evolved. Department of Botany also has a great history with its many dignified personalities as our heads of departments, and also we have very dignified teachers. We encourage our students and try to give maximum exposure to the new developments in scientific world and also give platform for scholarly interactions by arranging international and national seminars frequently, and also by arranging lectures and interactive sessions with great scientists and academicians. In 2020, due to the COVID pandemic situations, we may not be able to arrange such platforms, but due to many web-based platforms, we can manage to interact with illustrious personalities in the scientific world like Dr. Takada. Dr. Yoshikazu Takada, professor of Department of Botany and Molecular Medicine from US Davis School of Medicine, is a world-renowned scientist who was instrumental in the original identification of beta-1 integrins and also in molecular cloning of integrin, identification of several integrin ligands in 1980s. And this leads to the famous paper in Nature, fibronectin receptor structures in VLA family of heterodimers, which was published in Nature in 1987. And Dr. Takada identified regions of integrins that are involved in ligand binding and signaling by mapping functional antibodies using human mouse chimeras. And also, it is important in interpreting the crystal structure of integrin alpha V beta 3. He was instrumental in discovery of cytokines cross-talking with integrins through direct integrin binding. And he has many papers in many important journals like Nature and also like JBC. He has many papers in JBC. His works have built up the basis of the field of integrin biology and had impact in a wide variety of fields. And he is also a visiting professor of many universities with more than 6 patents to his credit. And he has total of nearly 220 publications and citations more than 20,000. And H index is 73, and as per the Google Scholar in September 2020. So even though he has such a busy schedule and he ignored the inconvenience of time zone difference, he agreed to spend his time and share his wisdom with us. So, Dr. Takada, we are extremely privileged and honored for your presence here. And on behalf of all those who are present here who are eagerly waiting for your lecture, I would like to welcome you wholeheartedly for this webinar and also to Indian audience. Welcome, sir. Next, next, I would like to welcome all my dear colleagues from the Department of Botany and also my colleagues from many departments like biochemistry, zoology, and many department faculties are here. And many faculties from other universities like Kannur, Cochin, MG University, and Calicut University faculties are also attending this webinar. So I would like to welcome you all for this program. And also, I would like to welcome many participants from other states. We have many participants from other states like Delhi, Tamil Nadu, Uttarakhand, Jammu and Kashmir, Karnataka. So I would like to welcome you all for this program, and especially I would like to welcome research scholars and my dear postgraduate students to this program. So once again, I would like to welcome you all for this program. And we can move to next part. That is, even though all of you might be knowing the biodata or history of his research, Dr. Takada's research, I would like to welcome my dear student to read the brief biodata of Dr. Takada. Please.
Okay. Can you see my slide?
Yes, sir. Just a second. One of my students will be reading your biodata. We can see your screen, but not the presentation, sir.
Okay. So here.
Just open the PPTs. Yeah. Now we can hear,
Okay.
sir. We
Okay.
can see.
Okay.
Sriraj?
Okay. Can you hear me? Can you see me? Okay.
Yes, sir.
Okay. Thank you for kind introduction, and then good evening, everybody. So I didn't know you are Department of Botany working on plants, and then I'm working on mostly in cells. So I will included a brief introduction to connect the plants and then animals. So this slide is including people involved in the work I'm presenting. Okay. So today I will show you some background on integrins and then short history of my study, and then that means history of integrin research, short history, and then move on to identification of new integrin ligand, what we are doing, and then the second work, identification of allosteric site on integrins, all about integrins. Okay. So this is a mammalian cells, and then can you see my here pointer? Can you see pointer? Okay. So this is the basement membrane. This is mammalian cells, and the integrins are located between cells and then basement membrane. So that's basically connecting cells and the basement membrane. So, and that is basement membrane is consisted of extracellular matrix proteins. Okay. So integrins over here, is here, this one. Yeah. I have to say this because when I presented this to a very senior biophysics professors, then question was, what is integrin? Yeah, okay. So I have to say this is integrin. And this part is cytoskeletal proteins, very complicated cytoskeletal proteins. And then this is the extracellular matrix. Basically, integrins are supposed to connect extracellular matrix molecule and the cytoskeleton here. Okay. So integrins, the role of integrins is that integrin connect the base ECM and the cytoskeletal proteins, and then that means integrins induce signals when cells are attached to extracellular matrix. So extracellular matrix proteins in mammalian cells, in the fibronectin, laminin, collagen are major ECM proteins. The integrin binds to these extracellular matrix proteins, and then that induce signal. Now, these signals are very important for growth, differentiation, morphogenesis, wound healing, immune response, and normal biological, physiological process, and also very important in the pathological process, in the pathogenesis of diseases, like cancer progression, metastasis, thrombosis, and the fibrosis. Are you familiar with these terms? That's my question. So, but probably this is generally enough for you, okay? So basically, integrins connect the cells and cytoskeletal proteins and the ECM, and then induce those signals upon binding to ECM. So it's a very important molecule, okay? So that's a major target of drug discovery. The many companies are working on that. Antibodies are smaller molecules. So I will talk about it in a little bit. So, okay, so question. So very general question. So if you look at the cells are binding to ECM, so what's going on? What kind of interaction is that? So this is very bright people, shown here. It's a Nature paper, Hirschbacher and Rusrati. And then this is a fibronectin molecule. It's a very big molecule, 200K kilodalton molecule. It's a dimer, so it's a 200 kilodalton here, and the one more chain here, and altogether 400K, huge protein. So they try to find out what part of fibronectin, ECM, is involved in cell binding, interaction with cells. So they chopped up into small pieces, and they found that this part is involved in cell binding, type 3 repeat 9 and 10 module. Okay, then they further, dissected this part, 9 and 10, and they found that small part of this here is very critical, or enough, very smallest component that supports cell adhesion. And then this contains motif RGD. This is a very famous classical RGD motif for cell interaction. And then what they found is that when RGD peptide is attached to beads, plastic beads, then a cell can attach to it. And then the control peptide that don't contain RGD or TGRD, G, don't support cell adhesion. The fibronectin itself can support cell attachments. So that's why they concluded that RGD peptide is critically involved. And interestingly, they found that this RGD motif is present in many extracellular matrix proteins, including fibronectin, fibrinogen, vitronectin, and so forth. Here RGD is conserved, distributed in many ECM. So that is a very important finding in 1984-85. And then why this is important? Because this triggered the extensive activity, drug discovery activity by pharmaceutical companies and then academics to find out a drug that broke cell attachment, because cell attachment is very critically involved in the disease, pathogenesis of diseases. Okay, so that is a 1984 paper was a small component. RGD is involved in cell binding in the ECM side, on the ECM side. Then the next work is also very important. They what component on the cell side, cell membrane side, is involved in cell ECM interaction. So they put the vitronectin, vitronectin or RGD peptide on the beads, and then labeled cell surface with iodine. So our cell surface protein was labeled with iodine, and then they made detergent lysate. So they incubated detergent lysate with vitronectin bead, and then eluted with RGD. Over here is RGD peptide, control peptide, RGD peptide. RGD peptide eluted two bands. 125. This paper says 125, 115. Two bands, and here. So this was a discovery of integrins. This is called at this time it's called vitronectin receptor or fibronectin receptor. So these two alpha beta structure is involved in cell ECM interaction. This is very important finding. First paper that appeared 1985. Okay, very cool work, smart. Okay, so then I started working on integrins, and then what we found is that another approach to find out what molecules are involved in cell ECM interaction, then is to make antibodies, monoclonal antibodies to cell surface. So we had the VLA is here antigen on T cell surface with unknown function. So we know this heterodimer alpha beta protein is present in cell surface, on the cell surface, but don't know what they are doing. Then we got the antibodies against integrins. Then what we found is that they cross-react with this our molecule. Also, then that it was a discovery of beta-1 integrin. So we identified VLA protein of unknown functions as integrin, cell surface, cell adhesion molecule. That is a pretty important discovery. And then here. Then we purified 1987, purified first integrin from human tissue. So I used human placenta. Okay, so I was working on before starting working on integrins. I was working on enzymes from animal hepatic tissue or kidney or some animal tissues. So I know we need a lot of protein source to begin with to purify very small amount of proteins. Then we got a human placenta from, I was working on in Dana-Farber Cancer Institute in Boston. Then next door was Brigham Women's Hospital. So then I got a very fresh human placenta from Brigham Hospital, Women's Hospital. Then I started working on that. So then within, I think, a week or so, using antibodies to these alpha chain, molecular antibodies to alpha chain, then we got alpha beta heterodimer using different antibodies. So beta is common, right? Beta is common. The alpha is size is different, but basically we got very similar pattern, alpha and beta. So then we did N-terminal sequencing of alpha chain here, alpha chain. But to our surprise, their N-terminal sequence is very similar. Okay, so they are kind of group of proteins, superfamily proteins. And at the same time, other group in Boston, Tim Springer's group, was working on another group of lymphocyte integrins. They don't call it integrins because we didn't have integrins at the time, namely integrins. Okay, so their N-terminal sequence is shown here. The another group in San Diego, La Jolla, purified beta-lectin receptor I mentioned before. And then this is a platelet integrin. Then their sequence was here. So when aligned, they are all similar. So we for the first time we learned they are superfamily of proteins. Okay, so that is 1980s, 87, something like that. Okay, so now what we know. This is summary, very simple summary. So now we have 18 alpha, integrin alpha, 18 alpha, and then 8 betas. Okay, beta 1, beta 2, beta 3, beta 4, blah, blah, beta 8. Then altogether, 25 different heterodimers with different ligand specificity. For example, these integrins binds to collagen, and then leukocyte integrin has different specificity to cell surface ligand, and then these beta 4 integrin, alpha 6 beta 4, and this integrin bind lamin. And then basically this group binds to RGD sequence or the ECM. So mostly, okay, so this is a group of superfamily of receptors that binds to different ECM and other ligands. That is what we know. And then many different cell types, at least many different cell types express different integrin patterns. They have at least one integrin, different combinations. Okay. So this is kind of complicated, one, but. So at least one, many different cell types express different integrins or cell surface, and then mostly they work on, they interact with different ligand, like laminin, collagen, and some fibronectin and some cell surface ligand, and so forth. Okay, this has been kind of a generally accepted concept, very basic concept. So now you can tell what is the integrin. Okay. So integrin structure was solved. We kind of know okay. So I forgot to mention. I cloned integrin alpha 2, alpha 3, alpha 4 sequence. It's a huge protein, and then we kind of know in the primary amino acid primary structure, amino acid sequence basically. Then 2001. First integrin structure came out from Boston again. Then this is a kind of cartoon based on the structure. So integrin is supposed to not have two conformations. So integrin is basically merge domain protein like this. And then integrin has a headpiece here. And the major conformation, difference to conformation is that one is kind of extended like this. And then another one is a bent structure. And the integrin has a transmembrane protein here, domain here, and the short cytoplasmic tail. So this tails are involved in interaction with cytoskeletal proteins. And then this part in the integrin headpiece is supposed to be ligand binding site. And then one model is that the bent form is not active, and then extended form is active. It's a very simply that is kind of widely believed. I don't believe too much, but that is a widely accepted concept currently. Okay, so then I start presenting my own work. We are currently doing. So integrins are receptors for extracellular matrix proteins, and then cell surface ligand. Okay, that is kind of right based on kind of historical perspective. So we first identified integrins as receptors for extracellular matrix proteins. That is kind of right. But because of that, the people still 2020 believe integrins are ECM receptors. That may not be correct. So we almost 15 years, 20 years almost. So we working on try to identify new integrin ligands using computer simulation. So virtual screening of protein data bank for new integrin ligands, okay, using docking simulation. Okay. So protein data bank contains more than 170 structures, crystal structures. And then, so I personally picked one by one, and then used docking simulation and screened potential integrin ligand. What we do is like this: small protein ligands, pick 100 proteins with 100 amino acid residue, okay, or less. Then using integrin headpiece, this contains integrin binding site, ligand binding site. and then of active form, active integrins. Active means, so some crystal structure was crystallized using RGD in it. Okay, so that means it's active. You know, definition is active. So we did a docking simulation and then repeat this 100 times simulation. Then each cycle, program pick a small lowest docking energy structure, and then this repeated 100 times, 50 or 100 times. Then this is a kind of cluster as a structure. Okay. So then what we got, for example, this is example in the case of FGF1, we first identified is a very nice first cluster with the lowest docking energy. So that is very likely is a structure poses of the FGF1 that binds to integrin. So this predicts that FGF1, for example, the growth factor, fibroblast growth factor 1, it's not the ECM, but that is predicted to bind to integrins. Okay. So then we did a, I told you, we have done, or I have done, 1,000, more than 1,000 checked 1,000 structures and found several, not several, but more than several, but mostly growth factors. Growth factor binds to integrins very well. So we found that, as shown here, FGF1, and then new regulin 1, this is also growth factor, EGF family member. And this is a chemokine, one of the chemokine, CX3CL1. And then this is the IGF, insulin-like growth factor. And this is a small protein, but not growth factor, but pro-inflammatory protein, secreted phospholipase A2 type 2A. Okay, so these proteins, for example, were predicted to be ligand for integrin. So what we found, kind of brief summary here, is integrin supposed to bind to ECM and the cell surface ligands, and this is important for normal biology and pathogenesis. But what we found is integrin also binds to growth factors like this, many growth factors. Not all, but many growth factors. And then what we found, so that the docking simulation predicted that growth factor can bind to integrin, alpha V beta 3, for example. But that at the same time, growth factor can bind to their own receptor, cognate receptor. For example, FGFR, IGFR, ARB3 is a new regulin receptor, EGF receptor, and then GPCR, G protein coupled receptors, like for CX3CR1, chemokine receptor. Then they make very complex integrin growth factor receptor complex. And this complex formation on the cell surface can be detected biochemically, and then they're critically involved in growth factor signaling. Okay. Furthermore, we generated a mutant that of growth factors that cannot bind to integrins, and then they were defectively in signaling functions. And then what we found is next we found, so that also predicted that these growth factor mutants are antagonistic. That is supposed to be dominant negative. Then that turned out to be dominant negative. Okay, so this right-hand side structure. So this is a kind of general summary, but I'll show you two examples now. Kind of surprising. So so I found that the integrin binds to FGF1. Then what's the function? What's going on? Why FGF5 have to bind to integrins? Then you look at the literature. Then I found that FGF and the integrins are kind of tightly related because FGF is a very potent angiogenic inducer. So when you include FGF to tissue culture, then you can induce angiogenesis, new blood vessel formation, basically. Then that can be blocked by antibodies against integrins. So this is in the first evidence that published in 1994 that FGF and the integrins are required for FGF signaling or growth factor signaling engine. This is called growth factor integrin crosstalk. Okay, so then this is a major topic at that time. New York Times first page type things. So because of this, hey, integrin antagonists can be used to treat cancers. Then that was a huge topic for pharmaceutical companies and the patient. So that was not that simple. Turned out to be more kind of complicated. Okay. So then, what is the current model of integrin cytokine growth factor crosstalk? Then, as shown here, integrins. How can I? Oh. So integrins are supposed to bind to ECM, right? ECM. Then growth factor binds to growth factor receptor. So then the two different signals, independent signals, merge together inside the cells and then induce signal transduction and the gene expression. This is the kind of current concept. Still 2020. This is 2007 version, but still very popular now. Okay. So okay, this is FGF. FGF is a heparin binding, very potent mitogen, and then induce proliferation, migration, differentiation. Very important growth factor. FGF is also a superfamily. So many superfamily, too many proteins, and but this is FGF1. Okay. So this is the same thing I mentioned. FGF1 is supposed to bind to integrin like this. I lost out here. Okay. Binds to this one. So what we have done next is that to make introduce mutation in here in FGF that can potentially block interaction. What happens? And then also this is a crystal structure of FGF1 FGF receptor complex. This is a crystal structure, and this part is a simulation docking model. And then when combined, what I saw this as kind of wow, this could be potentially important because FGF1 can bind to FGF1 receptor, FGF receptor, and the integrin at the same time, making complex. Oh, this could be very important. We should work on that. So then introducing mutations in the next. Yeah, then what we found is that one mutation in the interface, ELISA, shows that this mutant is very defective in integrin binding, but still binds to. So this is a SPR, Biacore surface plasmon resonance. Do you know that? This is a biophysical way to measure interaction Okay, so maybe you know already. So KD of integrin FGF1 interaction is about 1 micromolar. Is that a kind of reasonable affinity for integrin ligand? This is mutant. Mutant is very defective in binding. Okay, so we found this R50E mutant is very defective. So we analyze this mutant. So because that's defective in integrin binding, but not defective in heparin binding. FGF1, FGF, many growth factor binds heparin. So this mutation don't affect heparin binding or receptor binding here. Receptor binding is okay too. So but this mutant is very unique because it only block integrin binding. So what's the phenotype of this mutant? And then shown here, this is migration, cell migration. And this mutant is very defective in wound healing assay, cell migration assay. So we put the cells on the tissue culture plate and then remove cells using pipette tips. Then this wound is remove cells. Then is supposed to be healed. Here, this is wild type FGF1 induced closure, 60% in 24 hours. But this guy is very defective, like other control, very defective. And also DNA synthesis is also defective. Okay, so kind of defective in signaling. This is not really clear, but what we proposed at that time, this is a very old slide. So FGF binds to both receptor and then integrin at the same time, making complex. This is our idea, ternary complex. So current model is like this, and it's very different, right? So this is a model we still this that is correct. Then we can show this is the SDS page co-presentation of receptor or integrin with so when integrins are presented, that we can co-purify FGF receptor. And then when over here this side, and then when purify FGF receptor, then we can co-purify integrin. But this mutant, as predicted, is very defective in this ternary complex formation. So we have done same experiment using different cell types in both ways. Okay, so that shows that the integrin and then FGF receptor make complex biochemically. So yeah, this is biochemical. Then on the right-hand side, what happens when this this mutant is defectively integrin binding? Then this they cannot make a ternary complex. What happens? What is what can be predicted? So this we predicted that this mutant is antagonist, dominant negative antagonist. So dominant negative means, by definition, this mutant can compete with wild type and then suppress wild type signaling function. So is that really true? So next slide here. So this is a cell proliferation assay. Can you hear me? Can you hear?
Yes, sir. We can hear.
Questions? Okay, so left-hand side, this is cell proliferation assay. Then wild type can induce very strong cell proliferation here, but this mutant is very defective, not to zero, but they're very defective. Okay, so this is wild type is a mutant phenotype. Mutant is very defective in inducing cell proliferation or cell survival. So we put this low concentration and then added excess mutant. The question is that this mutant can suppress wild type's function to induce cell proliferation. Answer is yes. So when added, this mutant can suppress cell proliferation dose dependently. Okay, twice as much. Means less proliferation. And then this is a MTS assay. Is a kind of mitochondrial function. Proliferation assay using MTS. And then this is a BRDU DNA synthesis. The same thing happened. This mutant can suppress DNA synthesis induced by wild type. The conclusion is this mutant is a dominant negative mutant, is a decoy. Decoy means structurally very similar, but functionally defective, decoy. So this decoy may have potential use as a therapeutic. Okay, so we tested if that is really true. So we transfected, expressed this wild type mutant into cancer cells, colon cancer cells, and then tested if colon cancer proliferation can be suppressed. Answer is yes. So what happens is that this is a secretion into media. So this cell type, this mutant is secreted into media, culture media, okay, from cancer cells. They when injected into mouse, noodle mouse. What happens is that this is the wild type enhanced proliferation, and then this is a middle one is a control. I lost. Here. This is a control vector only, and this is a mutant. So mutant clearly suppressed cell proliferation of cancer cells in vivo and in vivo. Okay. So this is potentially, this might potentially have a therapeutic use. And then since FGF is a very potent angiogenic agent, angiogenic blood vessel formation, angiogenic, and then we tested if we can see that. So this is a kind of aorta, big artery from animal, rat, is removed, and then section of a big artery, our aorta, is embedded into collagen, and then FGF1 is added. So in the first, left, this one, you can see kind of a sprout, kind of angiogenesis, sprout from aorta here. And then mutant don't induce anything, nothing, no addition induce anything. Then when this was added in excess with FGF1, this can suppress angiogenesis here. So it's a potent angiogenic inhibitor. This is called aortic ring assay. So same assay, very similar assay using, this is a chick embryo. Fertilized egg is used to check angiogenesis. We put a hole in the fertilized egg, and they put a piece of paper with soaked with wild type FGF or mutant. Then count number of branching. You can see take picture and then count number. This is very tedious assay. But you can see here is a 5 nanogram per ml. You can see peak, maximum branching point formation, branching formation. Then mutant is very defective. Black column is very defective. When added, mutant is added together with the wild type, this clearly suppressed the wild type's function to induce branching. So this is again same conclusion. This is an anti-angiogenic agent. So we are trying to commercialize this one, but it's not successful. So if you are interested, we can do that. We have a U.S. patent, but it's not so easy to commercialize this. Okay, so second example. So this is the first example. I have several examples, but just one more. So virtual screening of protein data bank again identified chemokine is a small cytokine that is involved in cell migration. It's a major, also major drug target for disease, cancer and inflammation, chronic inflammation. So chemokine domain, chemokine structure is like this. Chemokine domain of this fractalkine CX3 CL1 is here. Chemokine domain here. So we made this one. Okay. So this is again I showed you chemokine binds integrin like this, but nobody really knows. So this is just a prediction. So chemokine is involved, and then conclusion is the prediction is that chemokine binds to classical ligand binding site of integrin, and then chemokine binds to chemokine receptor CPCR and then integrin at the same time. So that is what we know. We know kind of prediction. Okay. So we made again mutant that is defective in chemokine integrin binding. In this case, lysine residue 36 and 37, arginine 37 is involved in integrin binding. We know that. Okay, so then KD is about 10 to the minus 8. So this chemokine bind very, very strongly to integrins. So we have to measure this in the presence of calcium. Calcium is supposed to reduce integrin affinity. So, but still, if we activate, measure this KD at the very active conformation, active conditions, this goes up to minus 10. It's a very high affinity interaction. Okay, so we made this mutant. The mutant is very defective in integrin binding. Okay, but still this kind still binds to receptor. So we can co-precipitate receptor and then chemokine at the same time. So okay, but this mutant is very defective here. This is a control receptor binding defective mutant. So that's a negative control. So what, in conclusion, we made a chemokine mutant that is defective in chemokine integrin binding. So it's kind of, I just showed this one, just a part of this study. So what we found is that this mutant is again defective in chemokine signaling. So chemokine signaling in this case monocytic leukemia cells function is that to induce integrin binding. So this chemokine signaling is defective. Function signaling is defective. I don't tell you detail, but defective. So it's again dominant negative mutant. Dominant negative. We were able to generate dominant negative mutant by just removing integrin binding. This is an in vivo study, and then I don't show you in detail, but when this the drug is thioglycolate is injected into mouse peritoneum, cavity that induce peritonitis. Then a lot of leukocyte migrate into the peritoneal cavity. So we measure the number of leukocyte in peritoneal cavity, and then what we found is that this mutant fractalkine can suppress number of leukocyte recruitment in peritoneal cavity. So this means this mutant can suppress wild type's function again. So it's got a potential anti-inflammatory agent. So this is kind of summary. So we found many non-ECM ligand by using virtual screening of protein data bank, and then we found these guys, and then we show that they bind to integrin and the receptor at the same time, and then making ternary complex. This is in conclusion, this is very important. Complex formation is very important for the signaling function. So then what we found is that when we make a mutant that cannot be bind, cannot bind to integrins, that turned out to be antagonist. So this is a very nice strategy to make a potential antagonist. So I have I know many other growth factors, cytokine interact with integrin, and how to make antagonistic growth factors like this way. So this is just 2 examples. So I have several. Okay. So this is the same thing. So we can make a dominant negative mutant that cannot bind to integrins, and the dominant negative mutant cannot induce signal and act as an antagonist. So now we have several dominant negative mutant of FGF1, FGF2, IGF1, IGF2. IGF is a very important target in cancer. This is an important target in cancer too. And the CD40L is an important target for chronic inflammation. Integrin 1 is an important major target in breast cancer. Okay, so we have several mutants. Yeah. Yeah, so I told you summary. So virtual screen get this kind of idea that you can make a dominant negative mutant pretty, kind of directly, very simply and direct and logical way to access, to ask those questions. And then this is a very nice tool to identify potential new drugs. So then this is kind of second part. Okay, so slightly change directions. So first part of the talk. So I identified potential new ligand using active form of integrins. So we use active form of integrin, alpha V beta 3, for virtual screening. So and then at one time, I used the inactive form of integrins just for fun. Then what I found is that in this inactive form of integrins, chemokine, fractalkine binds to different site of integrin. So this is kind of second site, binding site. I didn't understand what is the role of the this interaction at the beginning. So why what is this second binding site? That's because crystal structure of integrin, RGD binding site is only one. Okay, so what is this second site doing? Okay, so this is a question, major question. Now, because this structure, this kind of relation, first ligand binding site, RGD binding site, and second binding site is very similar to allosteric site in enzyme or maybe hemoglobin. So I simply thought that this site, second site, may be involved allosteric activation. Okay, just the idea. Okay, this is what happened. So so docking simulation of this chemokine. This is active in the case of active. Sorry. Active integrins, chemokine bind here, red one. Okay, and then when I used inactive integrin like this structure, fractalkine bind here. That's a prediction. And then when superposed, this two structures kind of a mirror image. Site 1, site 2. I call this site 2. This is site 1. So mirror image. So this could be potentially important. So we pursue this direction further. And then in the last slide, I lost again. Okay. Yeah. So to prove this, site 2 is really present. I picked a sequence of around here, made a peptide, and then tested if this peptide can bind to fractalkine. Yes, it did. It bound to fractalkine, and then actually blocked function of this fractalkine. So then currently I know this is site 1, classical integrin binding site. This is bind. These are all the 5-nectin, 5-nogen. Many chemokine growth factors bind here. And then this is the site 2, over here, just like over here. So site 2, also many ligand bind site 2. Okay. So what I found is that integrin bind, chemokine to site 2 induced integrin activation. Then before going into that, integrin, what is integrin activation? So integrins are usually inactive in, for example, in platelet integrin, alpha 2 beta 3, platelet integrin is very inactive because otherwise platelet aggregate. It's very dangerous. Okay, and then induce thrombosis. Also, T cell receptor activation induce integrin activation. Then usually lymphocyte lymphocyte, leukocyte integrins are inactive. Otherwise, that activate very strong immune response. So integrins are inactive. Then integrin is supposed to be activated by platelet agonist, like thrombin. Thrombin activated platelet integrin, and at the same time, platelet is activated. And then start the thrombosis process. Also, T cell is activated by T cell receptor cross-linking, and then that induce many immune cell activation, many immune process. That is very important. But they have to be inactive. So and then something thrombin or T cell receptor cross linking trigger integrin activation. Okay. So and then second concept is that the integrin activation is accompanied by global conformational change. As I mentioned, that the integrin is supposed to be bent when they are inactive, and they are extended when integrins are activated. Okay. So that is the current concept. So this is kind of too much information for now. But as I told you, integrins are activated, and then when activated, they become extended. That's the current model, just a model. And then this requires, for example, thrombin stimulation, and that induce this inside signaling process here, and that induce is called clustering. Many integrin clustering, and then clustering ECM and clustered integrins interact with extracellular matrix. Extracellular matrix contains many integrin binding site. Okay, so that is a very activation, clustering, conformational change, and then keyword is that then thrombin stimulation, platelet activation. In this case, platelet. Okay. So so that contains, this is the same process, but integrins are usually believed to be only activated by inside-out signaling induced by thrombin or other things, platelet agonist. Okay, so now CHO cells don't contain anything, these components, protein kinase C or cytoskeletal protein kairin, or those kind of things. So they CHO cells, Chinese hamster ovary cells, don't respond to PMA, photopolyester, or thrombin, because CHO cells don't have thrombin. So when those components are expressed in CHO cells, then CHO cells can respond to all those stimuli. That is what happens. So this can be extensively studied, and then people can understand what is going on, what is missing in CHO cells, and so forth. But we recently found that this model is not perfect. So potential problem in this model is that ECM contains many integrin binding site. It's called multivalent. So they use multivalent ligand to detect integrin, for so-called integrin activation that actually integrin clustering, and then probably global conformational change. But they were not able to detect integrin affinity enhanced integrin affinity using monovalent ligand. So something is missing. So, sorry. So integrin, so-called integrin activation in this model is simply inducing integrin clustering and then global conformational change, bent form to extended, but not enhanced integrin affinity. So that is a problem. That's what I found. Okay. So yeah, so I maybe I can skip this, but even if integrin has low affinity, not affinity is not enhanced, but still can bind to ECM that has a multiple integrin bindings. So what's going on? So okay, then I realized allosteric activation by chemokine we found is important. Okay, so again, this go back to this slide. So we based on this finding, we tested if this cyto2 binding is involved in integrin activation. So next slide. So we used soluble integrin alpha V beta 3. Soluble integrin means no transmembrane domain or no cytoplasmic domain. But what we found is that so in this case, integrin ligand is immobilized to plastic, and we add a soluble integrin here, and then add the fractalkine. And then to keep it integrin inactive, we included the high concentration of calcium. This is a key. Calcium make integrins inactive. Okay, but even under those conditions, we were able to show chemokine can enhance integrin binding to ligand. So that means in solution, in cell-free condition, that means integrin can be activated from outside without inside-out signaling. So that is the point. Are you okay? Are you with me? Okay.
Yes.
So I don't see you, in this, Zoom form, I mean, Google Meet format. So I hope you follow what I'm saying. Okay. Yes or no?
Yes, yes.
Okay, so that's great. So soluble integrin can be activated directly by integrin. That is a kind of big surprise. So so we were able to show cell surface integrin can be activated by fractalkine like this. So I don't get into detail, but same thing. In calcium can keep integrin inactive, but this fractalkine can bind cyto2 and then enhance FITC labeled soluble ligand. Okay, so here. So without fractalkine, we don't detect very strong binding, but with fractalkine we do. Yeah. So as I already I showed you this slide, but cyto1 and cyto2 is totally different. And then what we know is that this peptide from cyto2 can bind to fractalkine. This is a peptide from cyto2 can bind to this fractalkine. And then in this case, this peptide can block integrin activation by chemokine. So that means this peptide shown sequence here is involved peptide and can interact with chemokine and then block it. So that means cyto2 is really involved in this allosteric activation. Okay. Yeah, so we found that several other proteins can also bind cyto2 and activate integrin. So this is one example. Another example is that this pro-inflammatory proteins. This is was originally found in fluid of rheumatoid arthritis fluid, the patient. So very pro-inflammatory protein. That's very well known. And then we found this also activate integrin, and allosterically activate integrin. Okay. So yeah, so same thing, same as fractal time activate in cell-free condition. That is important. So nothing to do. It doesn't require inside-out signaling. Okay. So what I'm proposing is that inside-out signaling can induce integrin clustering, as I told you, and then global conformational change. Probably yes, I don't know, but cannot induce enhance ligand affinity. Then, in contrast, this allosteric activation may not induce global conformational change, but still induce clustering. I don't know, but we haven't tested in detail. But this activation can induce or enhance ligand affinity to monovalent ligand. So that means directly enhancing integrin affinity. So that is missing in integrin activation, so-called activation by inside-out signaling. Okay, so those two pathways, two different mechanisms, complement to each other. Okay, so that I think is important. Okay, so this slide shows that. So question is that hey, there should be this what I'm saying is that two binding sites, cyto1, cyto2, right? So if there's two binding sites, and then PLA2, for example, this binds to both cyto1 and cyto2, then they should compete to each other. There is no evidence. What is the evidence that there is two binding sites? Very simply. Okay, so we tested this hypothesis. So we simply increased the amount of PLA2 added to the flow cytometry assay. Okay, so what happens is that over in here, so go up. So with low dose, this go up, and then at some point it go down again. So this here go up, integrin activation activities go up, and then go down. So why this happens? This is evidence that there is two binding sites. And then how do we interpret? So when at a low concentration, integrin is inactive, then what happens is that PLA2 bind only site to cyto2. Then open up cyto1. Okay. Then when with increasing concentration of PLA2, then the cyto2 eventually is saturated. Then this ligand start to compete with cyto1, ligand binding cyto1. So we used this fibronectin fragment.
Excuse me, sir. there is a problem. One of the participant is unexpectedly presenting. So, Miss Mukta Joshi, can you please stop presenting so that we can see the slides of Dr. Takada? Mukta, Mukta, please stop presenting.
Yeah. Okay, I did.
Yes. I think you have to again present now option.
Working?
Still not. Yeah.
Okay.
Yeah. Yes, sir.
Okay.
If
So
anybody
yeah.
can't
So
see the slides, please
huh?
pin to the slides. Okay, sir.
This
Please
slide, right?
continue. Please continue, sir. We can see the slides now.
Cannot see.
Yes, we can see.
Okay. So then start from here. So evidence. So there, what is the evidence that there is two binding sites? There are two binding sites, site 1 and site 2. So we just measured, increase the site 2, period ligand concentration. Ligand means activator concentration. So as shown here, at the low concentrations, this go up, integrin is activated, and they eventually go down. Why this happens? Explanation is that when there's two binding sites, site 1, site 2, and then when integrin is inactive, only site 2 is open. This is based on assumption that this is kind of allosteric enzyme. So site 2 is open, then activator binds site 2, and then integrin is activated. Then eventually site 2 is saturated, right? Then activator start competing with site 1. So that's why this will go down, right? Okay, makes sense. So this is evidence, clear evidence that there are two binding sites with a similar ligand affinity. Okay, so that is evidence. It's a pretty important finding. Okay, so site 2, we presented evidence that I presented part of it, and then several papers keep presenting, keep publishing papers, but still people don't believe it. Okay, so that's then last year one paper came out. It's a hydroxycholesterol. It's a very I didn't know that this is a pro-inflammatory lipid ligand for integrins. These people found that this hydroxycholesterol, pro-inflammatory protein, lipid ligand, bind to site 2, as shown here in this lower panel, and site 2, and they induce pro-inflammatory signal. So this I was very surprised, and I was very happy because other people showed that site 2 really exists, and that is connected to pro-inflammatory action. So ligand binding to site 2 induce pro-inflammatory signals. Okay. This is the evidence came from outside my lab. So this is, I think, a summary slide. So okay, so integrin activation by inside signaling is important, and then that requires several intracellular signaling component here, and that induce global conformational changes and the integrin clustering. That induced is important in signaling in cell ECM interaction, and then that I think by itself is a very important function. But something is missing here. Integrin affinity is not affected by intracellular inside-out signaling. But what we found is that many non-ECM integrin ligand binds to cyto2 and they directly activate, enhance ligand affinity to ligand binding affinity to monovalent ligand. So this, is not kind of contradictory. So they, two mechanisms can exist at the same time and they kind of complement to each other, independently working, probably, I guess. So, right? No. So we know that clustering is not induced, and so global conformational change is not induced. So we didn't detect it. Anyway, so the two mechanisms can coexist and complement to each other when cells interact with many other things through integrins. Okay. So I told you this summary. So I think this is the last slide. Okay. So this is all I have to say today. Maybe too much information, but you can tell what is integrin now. So what is so integrins are basically originally identified by receptor as receptors for ECM, but that is not whole story. So there are many non-ECM ligand present. We keep discovering. I have many things to do in my list, and then also integrin binds to classical ligand binding site. Then also there are another site, cyto2. Probably cyto2 is involved in integrin affinity enhancement to monovalent ligand. So this put this in your mind, and then we can discover or design new kind of drug antagonist. So we are now working on antagonists to cyto2 that may be as a potential therapeutics in cancer and then inflammation. Thank you. Thank you. This is all.
Thank you. So this is time for interaction. So if you have any queries, questions, doubts, please ask. Dear audience, dear participants. Sir, in the chat box, some people are asking some questions. Can you see it, sir? Hello, Dr. Takada?
Yes.
Can you hear? In the chat box, on the right side, there is a chat box. So some participants are typing certain questions there.
Just I don't see chat box. Just a moment.
Okay, I can read it, sir. No problem.
Okay.
So one question is from Prida Premjit. The question is, will you please explain how the integrins related to T cells?
Oh, T cell.
T cells.
T
cells.
Okay. T cell.
Yeah.
Integrin. Many integrins are leukocyte specific. For example, integrin alpha 4 beta 1, and then other one beta 2 integrins. 5 beta 2 integrins are uniquely expressed in leukocyte or lymphocyte. So they are involved in many immune responses by interacting with cell surface ligand. So yeah, that is very critically involved in immune response. That is what you want to know, or in more detail.
I think probably it might be okay.
Okay.
And Second, another question by same person, that will you please explain the importance of T cells, CD8 T cells, and CD4 cells in this COVID-19 pandemic? Is
For
there
COVID-19?
yeah? Is there any connection, or can you connect COVID-19 pandemic with the importance of T cells and CD4 cells?
Oh, I'm not really working on that. So
Yeah.
I'm not a good person to answer question. But what is important in COVID-19 is immune response to virus.
Yeah.
That is not only antibody, and then T cell cell-mediated immune response is also involved.
Yeah.
And the more importantly, cytokine storm is cytokine storm. So cytokine hyperinflammation is involved in the COVID-19. So that I think is a major cause of this. And then it's called cytokine storm. Extensively, many cytokines are, overexpressed, overexpressed in patient body, and that induce high inflammation. So, then antagonists to cytokines, growth factor cytokines, are potentially important. Antibody to TNF, or those are hyper activated cytokines are potentially useful. They are using it. So I don't know. So I don't know more than that. T cell, CD4, CD8, yeah, of course, involved in immune response to virus.
Yes.
But I'm not really a good person to answer that question.
Okay, sir. Then another question is, how integrins modulate cytoskeletal dynamics? Do they respond to apoptotic signals?
I don't know. Maybe, yeah, maybe important, but it's complicated. I don't know. Answer. So.
Okay, sir.
Sorry about that. I'm not really.
No problem, sir. Another question is, how is integrins play important role in signal transduction or in molecular trafficking for protein expression?
Oh yeah, so integrins, as I told you, integrin binding to ECM or cytokines induce many signaling intracellular signals. I didn't mention too much this time, but involved, and then that usually integrins ECM interaction or ligand interaction induce anti-apoptotic signals, like cell survival signals, probably mostly blocking apoptosis. So if you lose your cells lose ECM binding, so that induce apoptotic signals. And if you cells are connected to ECM, usually that block apoptosis. I think that is what my understanding.
Okay.
But it's a cell signal is very complicated.
Yeah. Any other questions from the participants? We haven't received any question in the chat box. So I think maybe we can conclude the session. So Anubama, is Anubama here?
Yes, ma'am.
Yeah. So we can move to next step, Dr. Takada. We would like to express our vote of thanks to you. So my student, Dr. Anubama, will be telling the word thanks. Anubama, please.
Okay, ma'am. Honorable honorable guest of honor Professor Yoshikazu Takda, and all other dignitaries of this webinar, well-wishers, staff and students. It's my privilege to have been asked to propose a vote of thanks on this occasion. I, on the behalf of the Department of Botany, University of Kerala and on my behalf, extend a very hearty vote of thanks to all those who have made this program a grand success. There is no magic to achievement. It's really about hard work, choices, and persistence. This is being proved by Professor Yoshikazu Takda through his spectacular achievement and outstanding contribution in Integrin biology. Even though Dr. Takda is a very busy person being director of the institute and research activities, he is willing to share some of his time and his valuable knowledge on Integrin biology. Thank you, sir, for enlightenment with us the novel concepts of Integrin, chemokines, and so on. You have explained the concept so easily to us. Thank you, sir. Sir, we students of Department of Botany and from the other departments, other participants from various parts of India are extremely happy and thank you for your time with us. We have been fortunate to be assisted by our HOD, Professor Dr. T.S. Sopna. You have T.S. Sopna, you have been a constant support and you have given us a lot of encouragement. Thank you, ma'am. I would like to thank all those eminent faculties of the department like Professor P.M. Radhamani, Professor Suhra Bibi, Professor Ganga Prasad, Professor E.A. Cyril, Professor S. Shiburaj, Dr. Bindu R. Nair, and Dr. Rajalakshmi for their involvement to take part in this event and making this a grand success. We are also extremely grateful to Dr. Reshmi Verma, who have introduced the speaker to us and helped to arrange the webinar. On behalf of the Department of Botany and my own behalf, thank you, ma'am, for all your efforts. Last but never the least, I thank all the research scholars and students for showing their interest in this program. I once again thank everyone for making this program successful. Thank you.
So let's wind up, Dr. Takada. So once again, I would like to express our thanks and gratitude to you, sir, for your presentation and your time. Even though it's in the early morning in your time zone, you are willing to give us the presentation and very interesting and informative talk. Thank you, sir, once again.
Thank you. Thank you for
Hopefully
inviting.
we will meet you in India and sometime. If there
Yeah.
is any chance, please come to Kerala and visit us. I would like to welcome you to Kerala, which is named as God's own country, which is a very beautiful state. So please do come to our state and university. If there is, after the COVID pandemic situation, if there is any chance, please do come.
Okay. Thank you.
Thank you,
I
sir.
hope you got something out of my talk.
Yes, 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 sir. Thank you. Thanks a
Thank you.
lot.
Okay.
Okay,
See you.
okay, see you, sir. Bye.
See you soon. Bye.
Bye.